methyl group on thymine, which is not present on uracil. Now, what you actually find is uracil is a precursor to thymine. So uracil is the first molecule, and then thymine is made from uracil. Now, to put this CH3 group on here, when you have a CH3 group, a methyl group, on a nucleic acid or on a nitrogenous base, it makes it much more difficult for a nuclease enzyme to attack the nitrogenous base. So if your DNA is methylated, and you have this on the DNA and when it's in its histone environment as well, if you have these methyl groups there, it's very difficult for enzymes to attack the DNA. So this is something else which provides, at the back, excuse me. Okay. So we have this thymine in DNA has a CH3 or methyl group. It makes it much more resistant to decomposition from these nuclease enzymes. So that makes DNA much more stable as well. RNA doesn't have the CH3 here, so it's going to be degraded much quicker by nuclease enzymes as well. And if you extract RNA in a lab, you have to do it very, very quickly because there's RNA nucleases in the environment everywhere. So if you want to extract your RNA, you can do it as quickly as possible in a really decontaminated environment. So RNA is very rapidly decomposed. And this is another reason why it doesn't have this methyl group here. Now, where's my sorry. My other interesting information for you here is for thymine to be created from uracil requires folic acid, so vitamin B6. So they found in pregnant ladies who don't have folic acid or deficiency in folic acid, they are much more at risk of having children that have leukemia. And the reason is because the thymine is not created, and uridine inserts itself into the DNA instead. So there's mutations in the DNA. So there's a link here between a causative link between not having folic acid in pregnancy and having childhood leukemia. Now, I've lost where I was now. Why? Did you not hear my explanation there? The link is because uracil is a precursor for thymine. If you don't have folic acid, which is required to create that methyl group from the uracil, you can't create thymine. So you have uracil in the uracil, so the nucleoside uridine in the DNA in place of thymine. So having these mutations can cause those causative effects. So these mutations can be linked to leukemia. So, where are we going? So these are nitrogenous bases. Now if we think back to, so sometimes you see different names. So you have your base, which is your nitrogenous base. Then a base plus a sugar becomes a nucleoside. And a base plus a sugar plus a phosphate group is a nucleotide. So if you see a nucleotide, that means it's your phosphate group, your base, and your sugar. So the name changes whether you look at the base, the nucleoside, or the nucleoside there. So this is just if you're reading and you think, well, I don't understand what's the difference between adenosine and adenine. So adenine is just the purine base. The nucleoside adenosine is your adenine plus your ribose. If you see deoxyadenosine, then that means that's your adenine and your deoxyribose base. If you also have phosphate group attached to that, then the name name changes. So this is just showing us here our nucleotides within RNA. Adenylate, which its full name is adenosine 5-monophosphate. Because we're having our 5' end of the phosphate here, and we have our adenosine base, our sugar, and our phosphate, and then we have the adenosine, guanosine, uridine, and cytosine. So these are just the names of the nucleoside. So as I said, thymine is more stable than uracil because it has this CH3 group in it. Now we'll find also that uracil, okay, it's not very chemically good way of saying it, but it is more friendly. Can we not chat in the back row, please? I can see I have eyes, I can have got ears. Okay, uracil. So if we have our nucleotides in RNA, we have guanosine, cytosine, uracil, and adenine. The complementary base pair for guanosine still remains cytosine, but adenine, there's no thymine there anymore, so it's going to bind with uracil. So that's its complementary base pair. But sometimes uracil can also bond with a G or a C to form pairs. And we're going to see this when we look at secondary structures in RNA. So when it's first produced, or when the RNA is processed, uracil will always be produced as a complementary to adenine. But when you have secondary structures and tertiary structures in RNA, you sometimes see uracil bonding with a C or a G. So, we've talked about the importance of the 2' on the ribose, or 2' OH on the sugar, which makes it a ribose. Now, I just want to talk a little bit about the 3' OH on the chemical sugar, and why is it so important? What's going to happen on our 3' end of our sugar? What links here? Yeah. This is where we're going to create our phosphodiester bond. So we have a 3' here. We have a phosphate here from another nucleotide. And this is where it's going to bind at this 3' end here. So we're going to have a condensation reaction, and we're going to create a phosphodiester bond similar to what you have in DNA. So, if we have a 5 to 3', and when we talk about transcription, this is going to be very important. The only place where you can have the phosphate group attached is going to be this 3' end here. The 2' ends here with the OH isn't reactive because it's due to the RNA polymerase which is required. The enzyme only works within this site here and not the 2' end of the OH of ribose. So we're always going to have our condensation reaction and the creation of your phosphodiester bond at this 3' end. So when we talk about our DNA and our RNA having direction, we're always going to add in the phosphate group of the next nucleotide on at this 3' end. It can't add on at the 5' end, because if you know the structure, it's not going to add the phosphate group on here. So, if you can see this very well, but this is just showing you your single-stranded is this additional OH group on the ribose makes it a much more bulky molecule, and it's very difficult for it to be stable as a double helix. So that's why generally RNA is single-stranded. And what we're going to see is it can bind to itself, and it can also bind to the DNA. But generally, RNA is single-stranded. So these are our predominant differences. DNA is double-stranded, generally RNA is single-stranded. We have a difference in the deoxyribose sugar for DNA, ribose for RNA, and the difference in the base here is we have uracil replacing thymine, so we have an additional CH3 on our thymine, which is going to, so this methyl group makes the DNA much more resistant to attack by nucleases. So, next we're going to talk about shapes of RNA. And conformation really means the shape and the structure of our RNA. Different types of RNA are going to have different conformations. There's a whole range of different names, like terpene loops, stem loops, tertiary structures, pseudoknots. These are all different ranges of RNA structure. Now, RNA is nucleic acid, but actually the folding of different types of RNA in different ways can produce or can make these certain types of RNA have an enzyme effect, so they have a catalytic effect. And these types of RNA are called ribozymes, because they're RNAs which have an enzyme effect. And there's very specialist ones which have specific duties to play. And when we talk about transcription later, not today, but next week, you'll see that some of them are involved in cutting up DNA and cutting up RNA, and putting that RNA back together again. So splicing is a cutting up effect. So the RNA can actually cell splice or cut things up and then put them back together again. So RNA is actually a very common one. So a primary structure of RNA would just be our single linear RNA. A secondary structure is when we start to have folding over, so when our complementary bases start to bond to one another. So here, a base pair complementation, uracil will bind with adenine, guanosine with cytosine, and this is bonded by hydrogen bonds, similar to how you would have on the double-strand DNA. And this causes strange types of loops and things within RNA. So this image here shows us the primary structure of an RNA molecule. And once we have such a secondary structure, we start to see, so very similar to proteins, we have a primary structure, a secondary structure, and there can be a tertiary structure as well. So the secondary structure is due to complementary strand binding. So we have a complementary sequence. Within one sequence, you can start to get those regions binding with one another. And these can cause all sorts of strange images, structures, loops within the RNA. Here is an example of a secondary structure of a type of RNA which is part of an RNA. So this is an RNA which works as an enzyme within E. coli. And by having this different type of structure has allowed us to have these different kinds of enzymatic effects. It's not a linear molecule anymore. So certain regions, when we have this binding in these kind of loops, and then this is a hairpin loop here, and this is a stem here. These are complementary base pairs from a linear structure coming and binding together. So, and what's happening is there's hydrogen bonds being created, or hydrogen bonds making this region stable. So, our RNA can go from a secondary structure in some cases becoming a tertiary structure where it folds even more into a much more complex 3D structure. And each molecule of RNA will fold differently depending on its base sequence. So we're going to talk about transfer RNA in a bit. And this is one example of an RNA which has a secondary structure, and and then it can fold further to have a tertiary structure, and the structure of this is going to be very important in the job that tRNA does. So, there's many types of RNA. Ribosomal RNA, messenger RNA, transfer RNA. They're the three that we're going to predominantly focus on. But we will find there's small nuclear RNAs in eukaryotes that are involved in processing messenger RNA, small nucleolar RNAs, which are in the nucleolus, involved in creating ribosomal RNA, cytoplasmic RNAs, microRNAs. The more studies that they do, the more types of RNA that they're now finding. So RNAs and chloride. So there aren't just three types of RNA. So we have to kind of get away from that thing that there are actually just three types of RNA. So the three that we're going to focus on today, because we're going to need this information for when we talk about transcription and translation. So we have messenger RNA, ribosomal RNA, and transfer RNA. So the name is hopefully going to help us with their function. So, messenger RNA. Anyone wants to tell me what messenger RNA is? Yes? In the eukaryotes. We don't have nucleus in the So it's going to be that message, it's that first signal when we have our DNA, and we want to create this protein, so we have to transfer our DNA, we have to change our DNA language into an RNA language, and this is the first point of difference. This is the messenger RNA which does this. So this is going to have this message from, so this is where our message from our DNA goes to our ribosome. So, messenger RNA is linear. It's going to be primarily composed of coding sequences. So a codon is a triplet of nucleotides which are going to code for specific amino acids. So our messenger RNA is going to have triplets of codons which are going to be encoded for particular amino acids. And this sequence is going to go to the ribosome. Okay, some messenger RNA also has non-coding regions. These are going to be untranslated, but these have specific functions as well. I can hear rumours, mumbling. Okay, so we're going to have untranslated regions as well. So we've got codons which are going to be translated. We also have untranslated regions. And we're going to see in a minute at some end, or the 5' end, of prokaryotic molecules on messenger RNA have particular sequences which are required for our ribosome to come in. If anyone's read their case study, it might be quite useful to pick up a little bit and listen. We will go over it next week as well. So, this is an example of a messenger RNA from a prokaryote. We have a 5' end, and we have a 3' end. And we know what these 5' end and 3' end means now. This is our end where our 5'-phosphate is, and this is where our last OH group is. Now, if we're going to continue transcription, which end would our next nucleotide go? Thinking about the structure of RNA, the 5' is phosphate on, the 3' is that sugar with the OH group there. Where are we going to put our next nucleotide? It's going to go attached at the 3' end. So we're only going to ever extend, or can extend, at the 3' end. Now, a messenger RNA doesn't just have a protein coding region, there's also going to be regions which are untranslated. But these have functions for the messenger RNA. A cell doesn't just have these sequences for nothing. Actually, all of them have a reason for being there. Now, you'll find in a bacterial cell of mRNA that we have a sequence which is generally the same in all prokaryotic messenger RNAs, and this is known as a Shine-Dalgarno sequence. Now, this has importance because when the messenger RNA is created, then a ribosome complex needs to come together for translation to take place. How does a ribosome know where to attach? At this sequence here, the Shine-Dalgarno sequence. What we'll find is the ribosome is another type of RNA. It's going to have a complementary sequence that binds here at the Shine-Dalgarno sequence. So messenger RNA has regions which are going to be required for protein coding and regions which are going to be untranslated as well. Now in eukaryotes, messenger RNA is different to prokaryotes. But one of the main reasons is, what does messenger RNA in a eukaryote have to do that it doesn't have to do in a prokaryote? It needs to leave somewhere. It needs to leave a nucleus in a eukaryote. It doesn't need to do that in a prokaryote. Prokaryotes also want to, or have to produce their proteins much faster. They don't generally have this extra function that messenger RNA has in a eukaryote. It wants to produce an mRNA, and transcription is going to happen, and practically after transcription, translation is almost immediate. Whereas in eukaryote, there's some additional things which are going to happen to our messenger RNA. So in a eukaryote, you have pre-mRNA and matured messenger RNA, and there's differences between the two. I'm just going to show you the image. In a eukaryotic cell, we have messenger RNA, or pre-mRNA here. We have regions called exons and regions called introns. Only some of that is required, so there's going to be some sort of processing which occurs. Can we have quiet, please? Shut up. Thank you. Whoever said shut up? I hope it's to me. But thank you anyway. So, you're still hear whispering going on. What we need to do is in pre-mRNA is going to have introns taken out and exons kept. So exons are going to be regions which are expressed. An intron is an intervening sequence which is going to be removed. Now what happens as well is that pre-mRNA then has a poly-A tail added. We're going to talk about some transcription in there, it's completely different. And it also has a capspaton, a methyl-guanosine cap, so a nucleotide cap on the end here. So this additional methyl group helps to stop decomposition. So this poly-A tail and this capping end here, and the untranslated sequences as well, are going to be involved in helping the mRNA have direction, leave the nucleus, and keep it from being degraded as well. So there's a difference between pre-mRNA and mature mRNA. Now, I'm running out of time, so I'm going to go faster. So we've talked about messenger RNA. Now we're going to talk about ribosome RNA. Now, one of the things that I think people sometimes get confused about are ribosomes just floating all the time within cytoplasm. Yes. Kind of. Now, ribosomes are much more complex than just a protein making factory, which is probably what some people may have thought. Yes, this is where we're going to produce our amino acid sequences, but a ribosomal complex is a combination of RNA and protein. Now, we're going to backtrack. We're going to start here with our assembled ribosome and come back to an unassembled ribosome. So, prokaryotes and eukaryotes have differences within their ribosomes. Now, in a ribosome, you'll see a number and then an S. This S is a sedimentation unit. So, you know when you did, cast your minds back now to what's it called now, cell biology, where you did ultracentrifugation and then you looked at different components of the different organelles and found different things due to the density of the organelle within the cell. Now, similarly, you can do experiments with ribosomes, extract the ribosomal RNA, and do different types of spinning, and at different times, depending on the density, you will get your ribosome out at different time. And it's a unit created called a Svedberg unit. So an S, so when you see, and the density, well, it's actually where it falls within the centrifugation depends on the structure of the ribosomal sequence. So how it folds together and the proteins involved. So we have two subunits come together to make an assembled ribosome, but here we see 50 and a 30 don't make 70. The reason is it's a unit of sedimentation, it's not just a unit of size. So by these two coming together, it changes the transformation of the structure and where it lies within the centrifugation. So we have a small subunit and a large subunit that come together to create an assembled ribosome. Now, within our large subunit, we have two regions of ribosomal RNA, and the small subunit goes one. They're going to come together to create this assembled ribosome. In the eukaryote, the ribosome is much larger. This has consequences because there are types of antibiotics. There's a prokaryote with a different size ribosome. There's different types of antibiotics which can be used to target a ribosome. So if you can create an antibiotic that targets 70 S ribosome, you can degrade all the ribosomal RNA within a prokaryote, but then it doesn't have an effect on the eukaryote ribosome because it's much larger. So this information is important. So, talk about that. This just tells you in a slightly easier format the different types of ribosomal RNA and their subunit locations. So, a small subunit ribosome and a large subunit ribosome due to the 70S ribosome. So, in prokaryotes and eukaryotes, it's different. So, our ribosome in itself has a structure. Believe it or not, okay, this is a cartoon image of a ribosome. We have a large subunit here and a small subunit here. And this is our messenger RNA, which has a sequence where our ribosomes first recognise to come together. And then we're going to have regions within our ribosome. There's an exit site, a site where we're going to make peptide bonds, and a site where we're going to put amino acids. So this ribosome comes together for translation, and we'll talk about this a lot more in translation. So we talked about messenger RNA, ribosomal RNA, and finally we're going to talk about transfer RNA. The tRNA is transfer RNA. Anyone want to tell me what this does? It brings in an amino acid. Yeah? How? Almost. So we're going to look. So there's a specific structure to our tRNA. So we said there's codons on our messenger RNA tRNA is going to have an anticodon to this, so it's going to have a complementary codon in it, which is going to match with that messenger RNA codon, and a tRNA, each tRNA with a different anticodon is going to have a different amino acid attached to it if it's a charged tRNA. So, often you'll hear this name, tRNA is a clover leaf structure. So often we don't really know what a clover leaf looks like. So, here is a normal clover leaf. Okay, if you're Irish or of Irish descent, a four-leaf clover is very lucky, but it's very rare. So, the clover leaf structure, normally three-lobed, actually tRNA is generally four-lobed actually, if I can put a four-lobed on there. But it just means it has a lobe structure very similar to a clover leaf. Now why have this strange structure? Structure relates to function. What we're going to see is there's a lobe here, a loop here, an anticodon loop here, where we're going to have an anticodon, and here at the top here, we're going to have an acceptor with an amino acid can attach. So depending on the anticodon that we have here, a different amino acid will attach here if it's charged. Now these other two loops here are involved in how the tRNA is going to come in and bind into that ribosomal complex. So tRNA also has some modified nucleotides, which this modification happens after transcription has taken place. So some of them are derivatives of uracil or similar to uracil, so uridine, so dihydrogen, uridine, there's some which are similar to thymidine, so there's some so these are rare nucleotides, but they are found within tRNA. Now, interestingly, these rare nucleotides are generally found at these lobe ends. So we have, it's called the DHU arm, that's because most commonly here is this rare nucleotide dihydrouridine. And it's T psi arm, the psi is pseudouridine. So there's different lobes here. What's important here is this anticodon loop at the bottom here, because this is what's going to match up with the complementary base pair of your codon of your messenger RNA. So this is our secondary structure. It folds even more with hydrogen bonding, so this is a tertiary three-dimensional structure of the tRNA. So, I don't have to go into that, just words saying what I've told you. So what happens here is we have our messenger RNA, which is here, it has a codon, a triplet nucleotide, an anticodon on our tRNA. Now here, if you look here, we have a 3' end and our 5' end. So if we unraveled all of this, it would make it linear. And at the 3' end, if the tRNA becomes charged, it will have an amino acid now. So whatever the codon is, it can have a corresponding amino acid. Now, there are more codons than there are amino acids. We will talk about this when we talk about translation, but what you'll see is on the tRNA, there's this thing called a wall position. So, sometimes this third triplet within our codon isn't really of importance. So, we could have an AGG or sometimes an AGC. So, sometimes the third complementary base pair doesn't match with that of the codon. And this is known as the degeneracy of the amino acid code. So if you have a codon UUU, that responds, or you're going to have a tRNA with phenyl aniline attached to the tRNA coming in on the messenger RNA molecule. But also, the codon UUC also is for the same amino acid. So we'll talk about this in translation more, but just telling you that we need to think about the structure of the tRNA and think about this anticodon and looping, why the tRNA has this structure. thing.